Good morning students. Today we will discussing about the chapter Biotechnology Principles and Processes.
It deals with the techniques of using live organism or enzyme from organism to produce product and process useful to human. We know that wine bread are produced using yeast, and yeast produce two enzymes known as inverters and cymers. These two enzymes are utilized for the production of wine, bread, etc. IVF, in vitro fertilization. It is another type of biotechnological process. Then the third one is plasmid technology. It is the advanced model of biotechnology technique in which wine bread production are present in ancient time. Also IVF is present in recent times, and plasmid technology is the advanced technique present in biotechnology. These are the principles of biotechnology. Genetic engineering. In genetic engineering, technique to alter the chemistry of genetic material, that means DNA or RNA. The normal composition of DNA and RNA are altered so that we can produce the desirable characters. Second one is maintenance of sterile or in vitro microbial contamination free condition. That means microbial contamination free condition or in vitro. In vitro means outside the body. In vivo means inside the body. So in vitro condition and microbial contamination free condition is necessary for the proper biotechnology. So these are the principles of biotechnology. Invention of plasmid technology jumped the biotechnology into a new era. That means plasmid are extra chromosomal. We can see in this picture, this is a bacterial cell. The bacterial DNA can be clearly observed, and also there is some circular structure other than the bacterial DNA can be seen. This circular structure is known as plasmid. Just we can look into the definition. Plasmid means it is the autonomously replicating circular extrachromosomal DNA present in bacteria. This helps for antibiotic resistance. That means in this picture you can see there is a circular structure. They are extrachromosomal. In this picture we can clearly identify that the bacteria have its own DNA, and these plasmids are extrachromosomal DNA and circular in structure. And the main function of plasmid in bacteria is this plasmid provide antibiotic resistance, specific antibiotic resistance for the plasmid. Stanley Cohen and Herbert accomplished this in 1972 by isolating the antibiotic resistant genes by cutting out a piece of DNA from a plasmid, which was responsible for conferring antibiotic resistance. That means these two scientists identified plasmid, these plasmids, and also they identified that these plasmids provide antibiotic resistance. The cutting of DNA at a specific location became possible with the discovery of molecular scissors or restriction enzyme. This, the invention of molecular scissors or restriction enzyme made the biotechnology field more popular. That means these enzymes can cut a specific site in genetic material so that we can cut any undesirable gene or desirable gene, so that this molecular scissor is very much famous in the field of biotechnology. Plasmid has an origin of replication. That means this origin of replication can be seen later. That the desired gene containing piece known as alien piece of DNA. Alien means other than that common, normal. That incorporate with the origin of replication site of plasmid. That means we can include this alien piece of DNA with the origin of replication. The incorporation of alien piece of DNA within the plasmid is called recombination DNA technology. That means there is a recombination taken place. The actual composition of plasmid has replaced with an alien piece of DNA. That is, the alien piece is the desirable piece, and so that this plasmid act as a new carrier, or and this technology is known as recombination DNA technology. Multiplication of desired gene containing plasmid in the host organism is called cloning. Cutting of desirable gene from the DNA is done by molecular scissors called restriction enzymes. These restriction enzymes are of two types: restriction endonuclease and restriction exonuclease. These two are included in the group restriction enzymes. Restriction endonuclease can cut inside a piece of DNA. That means they can cut inside, and restriction exonuclease can cut um, not in the middle, or it can cut in the corners. Then this cut piece is then incorporated with the plasmid, and they become sealed by another enzyme called molecular glue or ligases. These molecular scissors and molecular glue or ligase can collectively perform these functions, so that we can cut a undesirable piece from plasmid and add a desirable piece into this plasmid again, and sealed this portion by ligase, so that now the plasmid act as a new carrier, that the plasmid carry a alien piece of DNA or alien piece of desirable DNA. Plasmid act as a vector. This plasmid is incorporated into host organism bacteria, and it get multiplied in suitable medium. This multiplication can be discussed in the last section of this chapter. In this image, we can see a first complete plasmid in the first left corner, and they get one piece is cut from the original plasmid, and it get incorporated with a target gene. Here it is treated as insulin gene, and this cutting is done with the help of restriction endonuclease. Note that same restriction endonuclease must used in plasmid, and also when we use to cut the target gene also. Same restriction enzyme is required for cutting in two areas. So this digested restriction enzyme is incorporated with this plasmid. Then this incorporated piece is sealed by DNA ligase. Now in the right corner bottom portion you can see a new plasmid that carry the blue piece, that means the target gene, insulin gene. So now the recombinant plasmid carry a foreign gene called insulin gene, and this technology is known as recombinant DNA technology. The invention of restriction endonuclease, ligase, and plasmid made the biotechnology more popular and advanced. Then tools of recombinant DNA technologies. First one, restriction enzymes. The enzyme which cut a fragment inside a DNA is called restriction endonuclease, that we discussed earlier. That means enzyme can cut outside and inside. The inside cutting enzyme is known as endonuclease. First endonuclease was Hind second. Endonuclease cut a specific site known as recognition site. More than 900 restriction endonucleases are now known. Here is the naming of restriction endonuclease. ECOR1 is a restriction endonuclease. In this ECOR1, E stands for Escherichia, genus name of bacteria. That means E stands for the genus name of bacteria. Here it is Escherichia. CO stands for coli. It is the species name of that bacteria from this plasmid is obtained. That means this plasmid is obtained from a species known as Escherichia coli. Then R stands for strain in which bacteria obtained. First and the last one stands for order in which enzyme is isolated. Here one is the first. This enzyme is also obtained from the first order bacteria. Each restriction endonuclease can cut a specific site known as recognition site that we tell in the previous slide called palindromic sequence. Only restriction enzymes can cut a specific nucleotide sequence known as palindrome nucleotide sequence. Palindrome means we can say Malayalam, Malayam, and if we read from the back, Malayalam is also same Malayalam, and also like mom, MOM. If we read from the back, it is also mom. Such a sequence, read from forward and also backward same. Such a sequence is known as palindrome sequence. Here this type of sequence is also known as palindromic nucleotide sequence. Here it is GAATTC. From 5' direction to 3' direction, GAATTC is same in the first strand, and if we look into the second strand from 5' to 3' it is same as GAATTC. Only such a nucleotide sequence can cut by these restriction endonucleases. Second one, separation and isolation of DNA. The fragment of DNA is produced by using restriction endonuclease, and these fragments are separated by a process called gel electrophoresis. Gel electrophoresis means here this cut pieces are of different size and shape, size and weight, so that we can separate this restriction endonuclease this fragmented DNA by using a technique known as gel electrophoresis. Here agarose gel is utilized. Here we can see this process. We know that DNA is negatively charged, so it can move into positive portion. In the first figure there are 3 or 4 wells. 4 wells can be observed. The cut piece of DNA can be deposited in each wells and electricity is applied. When the power is turned on, DNA fragment migrate through gel because it is agarose gel, so it can move easily through from negative to positive charge. So the largest and more weight nucleotide sequence move slowly, and the smallest piece can move faster. This kind of methods we can separate the largest and smallest, and the smallest one get moved, collected and used for our purpose, or largest, if we require largest, we can collect the largest and used for our purpose. Here is the procedure. DNA is negatively charged, So it attracted towards the anode under electric power. We know that anode have positively charged through medium agarose gel medium. The separated DNA can be observed by staining it with ethidium bromide. We know that DNA has no color and also it is invisible by naked eye, so that it will require staining. Ethidium bromide is used here, then exposed to UV. Now we can see a bright orange colored band. That bands are DNA. Separated bands of DNA are cut from the gel piece. This step is known as elution. That means the non-visible DNA can be converted into visible by this process, and at last we cut these fragments from the agarose gel and moved into the next stage, and that process is known as elution. Vector. Vector means a carrier of something. Here, plasmid, bacteriophage, can replicate inside a cell without the help of chromosomal DNA. We tell something about plasmid, then bacteriophage. Bacteriophage means they are viruses that infect on bacteria, so that if we alter the genetic material of bacteriophage, we can alter with the desirable bacteria. Number of plasmid can increases from 15 to 100 or more in a cell by cloning or multiplication. We said that plasmid and bacteriophage is act as vectors, while we commonly used plasmid for our technologies because it is easy to handle and also non such pathogenic. Viruses, all viruses are pathogenic. If we alter the genetic material of bacteriophage, it may be converted as mutated form and may cause diseases. While in the case of plasmid and bacteria, it is comparatively easy to deal. More than 15 to 100, 15 from 15 to hundreds of plasmids are present in a cell and can be increased by process called cloning or multiplication. These are the features of a vector. Origin of replication, commonly known as ori. It is the replication starting site and also control the number of copies of the vector. We earlier discussed that our desirable gene is incorporated with the origin of replication. Second one is selectable marker, which helps in identifying and eliminating non-transformant and selectively permit the growth of transformant. Example, ampicillin resistant, tetracycline resistant. This selectable marker can be discussed in the later stage. Then cloning site. This is the site for joining the desirable gene to the vector. There are various cloning sites present in a plasmid like BAM H1, SAL1, PVU1, etc. Here is a picture of plasmid, E. coli cloning vector known as PBR322. PBR322 is the name of this plasmid. In this plasmid, you can see the origin of replication, then ampicillin resistant gene, tetracycline resistant gene, and various cloning sites like PST1, PVU1, EcoR1, CLA1, Hind III, BAM H1, SAL1, etc. are the cloning sites. And this is a typical example for a cloning vector called plasmid. Its name is PBR322. Insertion inactivation. if a plasmid containing both the genes, that is ampicillin resistant and tetracycline resistant, that means in this picture you can see in this plasmid there are both ampicillin resistance and tetracycline resistant genes are present. So this plasmid can be taken in our studies, and we remove the tetracycline gene and insert an alien piece of DNA desirable. So we must use same restriction endonuclease for removing this tetracycline resistant gene and also removing this alien piece of DNA from the host. That means the tetracycline gene in the right side red colored portion can be removed and insert an alien piece. That means this plasmid is incorporated with a new gene, and then the plasmid is placed on a medium containing ampicillin. Now that means now we place this plasmid in a ampicillin placed in an ampicillin medium containing ampicillin containing medium. Ampicillin containing medium, the inserted and non-inserted will be survived. That means if we remove the tetracycline gene from this bacteria and alien piece is inserted, and this plasmid is again inserted into a bacteria, and that bacterial cells are placed in a medium containing ampicillin. So now also that bacteria contain plasmid, and that plasmid contain ampicillin resistant gene, so that that bacterial cell can survive. So we can look into next stage, that is if we place them in tetracycline medium, the same, that means the tetracycline gene is removed and inserted an alien piece. Now that bacteria only, or that bacteria or plasmid that only contain ampicillin resistant gene, then we place them in tetracycline medium, then non-inserted will be survived and other become died due to the absence of tetracycline resistant gene. That we already said about. The plasmid provide antibiotic resistance for bacteria. Now we remove one of the antibiotic resistance from this plasmid, so that when such antibiotic is utilized, that bacteria can't be tolerate in such a condition. This process of inactivation of gene by inserting of foreign DNA is called insertion inactivation. That means the process of inactivation of gene by inserting of foreign DNA is called inactivation. In this case, we remove tetracycline gene and insert a foreign gene here, so that that tetracycline gene get inactivated and it activate a foreign gene in that plasmid. Here we can say that it is an insertion inactivation. Beta galactosidase inactivation of recombinant plasmid is now used. Uh, by such a process, we can alter the pathogenic and harmful effect of some organism into human friendly or animal friendly or plant friendly in nature. Agrobacterium tumefaciens. This is a type of bacteria which can cause cancer tumor in plants. It can be deactivated and converted into vector for desirable gene. That means we remove the cancer producing genes from these agrobacterium tumefaciens and incorporated that area with a desirable gene, so that when these transgenic agrobacterium tumefacients is infected with that plant, they do not cause cancer, and the incorporated gene, that means the desirable character gene, is expressed in that plant. So we convert a pathogenic organism into good one. Like the same way, there is a possibility of converting non-pathogenic organism into pathogenic also. Such a process of converting non-pathogenic organism into pathogenic and dreadful organisms is known as constructing of bioweapons. And retrovirus also can convert to good by altering its cancer causing gene to a good desirable gene, and later can introduced into animals. Retrovirus means reverse transcriptase ability ulla വൈറസുകൾ. That means reverse transcription means the process of conversion of DNA, production of DNA, sorry, RNA from DNA is known as transcription. Reverse transcription means the process of conversion from RNA to DNA. Such reverse transcriptase viruses can produce DNA utilizing RNA. Normally, the process is from DNA, RNA is produced, but this type of organism can convert or produce DNA from RNA. Plasmid. It is also a vector in which cos stands for cos site of lambda phage and MID stands for plasmid. That means it is the collaboration of both lambda phage and plasmid. Then bacteriophage that we discussed earlier. That means bacteriophage is also can be utilized as plasmid. Cloning vector. But the most commonly used vectors are plasmids. We move to transferring the recombinant DNA into host technology. Bacterial membrane is not permeable. We know that bacterial membrane has cell wall made up of N-acetyl glucosamine and N-acetyl muramic acid. So they are not permeable to plasmid. So first of all, we must convert this bacterial cell wall into permeable. For that, we must treat it with divalent cation. Here we use calcium cation. So this divalent cation made pores on it. Then it treat with ice. Then suddenly transferred to 42 degrees Celsius and called heat shock. Then again back to ice. That means first made pores, then transferred to ice, then suddenly transferred to 42 degrees Celsius, then again suddenly transferred to ice. So that made a shocking nature. So at a time, this plasmid is taken by this cell. In another method known as micro-injection, this micro-injection method and gene gun method etc. are the recent and modern techniques. And the earlier discussed heat shock method is the ancient or the first, not very modern but recently used. Recombination DNA is in microinjection. Recombinant DNA is directly injected into the nucleus of an animal cell. In another method suitable for plant cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA in a method known as biolistics or gene gun. These micro injection and gene gun methods are adopted as advanced, and the last method used is disarmed pathogen. In such a method, the plasmid is vector which allow to infect the cell, transfer the recombinant DNA into the host. That means the disarmed means non-pathogenic non-pathogenic agents are allowed to infect on bacteria and transfer the plasmid into bacteria. These are the processes of recombinant DNA technology. First one is the isolation of DNA. Second one is the fragmentation of DNA using restriction endonuclease. Third one is ligation of DNA using ligase. Fourth one is culturing the DNA in large scale. First process is isolation of DNA. To isolate DNA, we must first degrade the outer cell wall by treating it with lysozyme. In the case of bacteria, we know that the bacterial cell can be destructed using lysozyme. In the case of plant cell, we can use cellulase because plant cell wall is made up of cellulose. In the case of fungal cell wall, we can use chitinase because chitin is the material that the plant cell is made up of. Then the DNA is situated in nucleus and there also contain RNA, protein, etc. These are also removed by RNase, protease, etc. And no DNAs was used because if we also use the DNA are also disintegrated. Then the non-disintegrated DNA, disintegrated RNA, protein are treated with chilled ethanol. Fine threads can be observed in ethanol and they are DNA. That means the outer cell wall of fungus, fungus, plants, bacteria are made up of cell wall. In the case of animal cell, there is no cell wall. So we must first treat it with various chemicals according to their composition and destruct it. Then the nucleus can be destructed using nucleus, RNAs, protease, etc. And also these DNAs can't be not be used because that DNA is required. Then the DNA cut using restriction endonuclease. DNA can be cut into using restriction endonuclease. Later, this required piece can be amplified using PCR, that means polymerase chain reaction. We know that this isolation of DNA is a complex procedure and required very attention and carefulness. So the constructed DNA piece can be multiplied using a technology known as PCR. This PCR is widely used in the field of medicine, forensic science, etc. In the case of forensic science, only a few samples are obtained. In order to ensure the maximum number of copies, the forensic scientists amplified these collected samples using PCR. We can check how this PCR is working. The full form of PCR is polymerase chain reaction. Three stages are present in a cycle of polymerase chain reaction. They are degeneration, annealing, and primer extension. It can be explained by a diagram. Here we know that DNA is a double-stranded structure and each strands are connected by nucleotides. First of all, we in the first stage, that means denaturation. In the stage of denaturation, application of heat is the main procedure. That means 94 degrees Celsius to 98 degrees Celsius, so that this double stranded DNA get converted into single stranded DNA, and that process is known as denaturation. In the second stage, that is annealing. At that annealing stage, the temperature lowered gradually. That means 55 degrees Celsius to 70 degrees Celsius, and so on. And in annealing, the main procedure is the addition of primer. In that primer is a small stretch of RNA that attached with each strands of DNA in the 3 dash end, while the primer at primer's 5 dash end. We can clearly see in this picture. In the 3 dash end, the primer get attached. The next stage, that is primer extension. Here for this primer extension we must add a polymerase known as Taq polymerase. The full form of Taq polymerase is Thermus aquaticus. Here we know that the temperature is approximately or averagely above 70 degrees Celsius or 50 degrees Celsius, or and so on, and high temperature is also present. In such a condition, normal polymerase enzymes can't work. At that situation, we construct or not construct, we isolate an enzyme from a polymerase enzyme from an organism known as Thermus aquaticus, so that this Taq polymerase can withstand in high temperature, and this Taq polymerase collect the deoxyribonuclease. That means these deoxynucleotides from this pool and construct the strands and construct the entire strands based on the template strand, so that from a single template DNA, two copies of DNA can be produced at the first cycle. Again, from these two DNA strands, four DNA strands can be produced. So and so on. There is a chain reaction taken place, and around 30 cycles, 1 million copies of DNA can be produced. First stage is degeneration. That means double stranded DNA becomes single stranded by heating in the machine. And the second stage is annealing. First stage is not degeneration, it is denaturation. Second stage is annealing means the addition of primer over each single strand of DNA. Primer is a small stretch of RNA. Then the third stage is extension. The addition of DNA polymerase, Taq polymerase plus deoxynucleotide is the third stage. Sorry for the trouble. First stage is denaturation, not degeneration. First stage is denaturation. Taq polymerase is isolated from Thermus aquaticus, which can work in heat condition. This polymerase can work even in heat condition. In 30 cycles or more, the DNA become amplified into million copies. Then various methods like heat shock, micro injection, gene gun method, etc. The incorporated plasmid can be entered into the host organism, that is bacteria. Here we move to selectable markers. It can be explained using an example. One cell contains ampicillin resistant gene containing plasmid. That means there are two cells. In the first cell there is no alteration taken place. Both ampicillin and tetracycline genes are present in that plasmid. In the second cell, the plasmid does not contain ampicillin gene. That means that ampicillin gene is removed and an alien piece of DNA is incorporated. Then both the cells are placed in agar plate containing ampicillin. Then the cell which contains the plasmid, ampicillin carrying plasmid, survives. And the other cell which does not carry ampicillin resistant gene dies. That means we can get an indication. One has ampicillin resistant gene and other has no ampicillin resistant gene. In that indication, we can clearly understood that the non-ampicillin containing gene is the transformant or transformed one. In such a condition, the ampicillin resistant gene is termed as selectable marker. Here we utilized this ampicillin resistant gene for identifying whether it is transformed or non-transformed. So we can call this ampicillin resistant gene as selectable markers. So the ultimate aim of this plasmid technology is for to obtain foreign product. So we must move into this foreign product production process. The ultimate aim of transfer of plasmid is an cell is to produce protein inside the cell. That means it's a desirable protein. In economic point of view, increased production made benefit like the insulin production, other hormones such as insulin or any other medicinal protein, etc. In order to increase the production, we must change the lab scale to industrial scale. This plasmid technology and construction of desirable gene plasmid and cells and their culture etc. are done in laboratory condition. Only after the successful completion of all the mentioned procedures, an organism is labeled as a transgenic animal or transgenic microorganism. And this transgenic microorganism can be utilized for further processes. For the industrial production, the bioreactors or biofermenters are used. Bioreactor or biofermenter are an instrument. A bioreactor provides optimum condition for the growth of cells like oxygen, nutrients, temperature, pH, etc. And also the impeller inside the reactor provide uniform distribution of microbes and nutrients over the entire fermenter. That means the microorganism can spread over the large bioreactor and the impeller provide the proper uniform distribution of the microbes and all the desirable characters are provided inside the bioreactor. Normally a stirred tank fermenter is used for this purpose. Stirring means just stir the all content for the uniform distribution. That stirring mechanism is controlled by a motor. After the completion of all the processes, the product that obtained as a result of gene expression, that means protein. For example, in the case of insulin, the insulin that now available in our market are produced through these procedures. Expression protein inside the fermenter get isolated, purified, etc. and that process is known as downstream processes. The fermenter contain so many unwanted things like the remnants of food, nutrients, pH material and the dead microorganism etc. We only require the protein of desire. That means the desired protein, desired product. That isolation of the desired product, purification, Such process is a complex area, and that process is known as downstream processing. Here we can see a biofermenter or a bioreactor, and the agitation system means it is the impeller. It controls the impeller. Impeller agitation system is the motor. Then feeding pump. The through the feeding pump, the medium containing all the nutrients etc. are provided. Then aeration. Proper aeration means bacteria, aerobic bacteria required proper oxygen. Through aeration we provide oxygen. Then reactor tank, then submerged aerator, then sensor probe, sensor for which product is depleting or which product is available more is can be sensed by these sensors. Then thermal jackets. Due to the bacterial metabolic activities, there is a chance of production of temperature. So in order to overcome these difficulties, thermal jackets are provided. Then the effluents can be removed using a effluent controller. This is a kind of biofermenter, or also known as bioreactor. I hope all the topics that we discussed is understood.
Thank you for watching this video. We can see in the next class with a new topic.